Hi friends, welcome you all. So in this video lecture, we'll going to start different method of preservation of foods to avoid spoilage condition. And uh, in this lecture and upcoming lectures, we'll study so different method. These includes physical method as well as chemical method, as well as different kinds of radiations that are used for preservation of food. So first we're going to study physical methods. In that we'll see thermal processing. So we know that heat treatment we usually employ for sterilization in case of microbes. So if we want to eradicate or complete we want to complete removal of microbes, then we use heat as well as steam under pressure. So this type of sterilization is very effective as far as the other means are concerned. So here in case of food also, in a previous lecture in case of milk we have seen that we use pasteurization techniques. In that there are different modes of pasteurization we have studied, say for example a high temperature short time method or ultra high temperature short time method. So in this way in case of food also the same principles are applied here. So we'll see heat kills microorganism by changing the physical and chemical properties of their protein. So how heat kills, it changes the physical as well as chemical properties of their protein. So microbes they get denatured. So when heat is used to preserve food, the number of microorganisms present, that is called as microbial load, is an important consideration. So the extent of heat and time of application of heat is pretty much depend upon the microbial load that is present in food, and various types of microorganisms must also be considered because different levels of resistance exist. For example, bacterial spores are much more difficult to kill than vegetative bacilli. So this is the general scenario in case of spores, that they are pretty much resistant to heat, and even after higher temperatures the spores they get still remain as compared to vegetative cells. So vegetative cells of bacteria they get destroyed by heating. So in addition, increasing acidity enhances killing process in food preservation. So first we're going to study high temperature. So three basic heat treatments are used in food preservation. These are pasteurization, in which foods are treated at about 62 degrees Celsius for 30 minutes or 72 degrees Celsius for 15 to 17 seconds. Then next, hot filling, in which liquid foods and juices are boiled before being placed into containers. So this is again temperature based method. In that liquid foods and juices they are treated through boiling before being placed into containers. Then next is steam treatment under pressure. So steam under pressure, you have studied in principle of autoclave, that when steam you apply pressure to the steam, so it is very effective in eradication of different means all kinds of microbial flora. So such as steam under pressure use, such as use in canning method. Then heat resistance of microorganism usually expressed as the thermal dead time. So the thermal dead time is the concept which is basically related to heat resistance of microorganism and the time necessary at certain temperature to kill a stated number of particular microorganism under specified conditions. So this is what is meant by thermal dead time. So it is the heat resistance of microorganism, and it is the time necessary at certain temperature to kill a stated number of particular microorganisms under special conditions. So here in case of pasteurization, it is the process of heating food usually in liquid to or below its boiling point for a definite period of time. So simply in pasteurization what we do, we provide heat for specific time, and there are different combination of heat as well as time, and the purpose is to destroy all pathogens. to reduce number of bacteria and to inactivate enzyme and extend the shelf life of food product. So overall these things we achieve by the process of pasteurization, and pasteurization treatment is able to kill most heat resistance nor spore forming organisms like mycobacterium tuberculosis, then Coxella burnetii, etc. Then food with a pH of less than 12.6 such as milk and spaghetti sauce can be pasteurized. So permanent stability that is a shelf life of about 2 years is obtained with foods that can withstand prolonged heating such as bottled juices. So there is a greater loss of flavor from food that are exposed to longer time temperature relationship. It is obvious that longer time treatment affects the quality of food, and therefore temporary stability, that is limited shelf life, is only obtained with some foods where prolonged heating would be destroy its quality. So these foods such as milk usually require subsequent refrigeration. So high temperature short time EST or ultra high temperature UTH process have been developed to retain food texture and flavor quality parameters. So pasteurization is not intended to kill all microorganisms in the food. Instead, pasteurization aims to reduce number of viable pathogens. So basic aim of pasteurization is to remove pathogens so that they are unlikely to cause disease. So pasteurization involves a comparatively low order of heat treatment and generally at temperature below the boiling point of water. So eating may be done by means of steam, hot water, dry heat or electric currents. Okay, so pasteurization is carried out mainly by means of steam, then simply by hot water or dry heat or electric currents. So products are immediately cooled, and desired pasteurization can be achieved by combination of time and temperature, such as heating a food to low temperature and maintain for a long time, that is LTLT, means up to 68 degrees Celsius for 30 minutes. So it is low temperature high time combination, or heating of a food to high temperature and maintains for short time, that is the STST, it is up to 72 degrees Celsius for 15 seconds. So these are different combinations of time and temperature are used for pasteurization of foods. Pasteurization is used when more rigorous heat treatment might be harm to quality of food product as the market milk and for the main spoilage of organisms which are not heat resistant, such as yeast in fruit juices, and it also kills the pathogen. So this process of pasteurization, it is not carried out for preservation or processing of many foods, but it is carried out in some foods, especially in liquid foods, in milk as well as milk related other products and some juices, and in case of some beverages it is carried out. So this is how batch pasteurizer look like, and you can see in this diagram the picture showing batch pasteurizer. Okay, so now we see ultra heat pasteurization. So in this process milk is heated at 120 to 138 degrees Celsius for 2 to 4 seconds and followed by rapid cooling. So this is how pasteurization is achieved, retaining the default quality of milk. So it is again a time temperature combination, and a high temperature for shorter time and rapid cooling. So this combination achieves the greater efficiency of pasteurization, as well as it retains quality or nutritional conditions of food. Especially this type of treatment is utilized for milk as well as milk related other products, and this treatment kills all spoilage microorganism. So ultra high treatment pasteurized milk is packaged aseptically, resulting in shelf life stable product that does not require refrigeration until it open. So this kind of with these kinds of treated milk, so we can put milk for some longer period, and it does not require any kind of refrigeration, and until and unless we open it, it cannot be, it cannot get spoiled. Then heat resistance of microorganisms and their spores, so it is expressed in terms of their thermal date time. So what is meant by thermal date time? So it is a heat resistance that is shown by microbes and their spores. Now it is the time taken to kill a given number of microorganisms or spores at certain temperature under specified conditions. So this is how we can define thermal death time. So it is the time taken to kill a given number of microorganisms or their spores at certain temperature under specified conditions. Then thermal death point. So see, these are two different things. Whether first we have covered thermal death time, and this is thermal death point. So it is the temperature necessary to kill all the microorganisms in 10 minutes. So here what we do, here what the tenure or the time we kept fixed, it is for 10 minutes, and whatever temperature required, it is considered as an thermal death point. So it is basically a temperature necessary to kill all the organisms in 10 minutes. Means this process is carried out at fixed time, and the temperature may fluctuate. It may be 60, it may be 80, it may be up to 120, etc. So heat resistance of different microorganisms is different. Obviously, this depends upon pretty much properties of membranes, and microorganisms are more heat resistant than their spores. So heat resistance of vegetative yeast is up to 50 to 58 degrees Celsius in 10 to 15 minutes, and ascospores is 60 degrees Celsius for 10 to 15 minutes. However, yeast and spores are killed by pasteurization. So heat resistance of microorganisms, so there are some heat resistance modes also, for they can even withstand 60 degrees Celsius for 5 to 10 minutes, and asexual spores, they are again more heat resistance than ordinary mycelium and require temperature of 5 to 10 degrees higher for their destruction. So aspergillus, mucopenicillium are more resistance than yeast, and heat resistance of bacteria and bacterial spores is different. So cells of high in lipid content and capsule containing bacteria harder to kill. So as I told you before that killing and heating of means killing property of microbes is pretty much depend upon membranes. So higher the lipid content in membrane and capsule present in, so higher their resistance to heat they will be. So higher the optimal and maximal temperatures for growth, the greater is the resistance to killing. Then heat resistance of enzyme. So most of the food and microbial enzymes, they are destroyed at almost 80 degrees Celsius, and some hydrolysis will retain substantial levels of activity after ultra high temperature treatment. So bovine phosphate, if present is in processed milk, it indicates that milk was not properly pasteurized. So not only microbes they get killed, but enzymes they produces, say for example, they may be toxins or other toxic chemicals, so they also should get destroyed by these heating procedures. Then this is again important topic, that is d value. So it is the decimal reduction time. Okay, so what is this? It is decimal reduction time, or the time required to destroy 90% of the organism. So what is meant by d value? So simply it is the time that is required to destroy 90% of the organism. Means this is at this value almost 90% of the population it gets reduced, and it is denoted by symbol d. So mathematically it is equal to reciprocal of slope of survival curve, and it is measure of death rate of microorganism. Okay, so this term is especially related to death rate and death of microorganism, and it is expressed in case by symbol d. And when d is determined at 250 degree Fahrenheit, it is expressed as dr. So this is standard 250 degree Celsius Fahrenheit. So when we express d at this particular temperature, so it is referred as dr. Okay, so this is d value of organism. So here in x-axis you can see different time intervals of heating. In that you can see 5 hours, 5 minutes, 10 minutes, 15 minutes, 20 minutes, then 25 minutes and 30 minutes. And here you can see at y-axis the survivors, means after heating to these for longer time, how many microbes they are survive at last. So obviously very few will survive, and most of will they get dead. So this is the reciprocal of survivorship curve, and this d value is better expressed as 250 degrees Celsius Fahrenheit. Okay. Then next, z value. So it is refers to degree of Fahrenheit required to reduce TDT tenfold. Okay. So what is this meant by F value, Z value? It is the temperature. Okay. So what is it? It refers to degree of F. F means what? It is degree of temperature required to reduce thermal dead time 10 volts. Okay. So mathematically this value is equal to reciprocal of slope of TDT curve. So here also, here what we calculate? We calculate temperature, and here we calculate the time. Okay. D value we calculate reduction time, and at Z value we calculate a specific temperature to reduce thermal dead time by 10 volts. Okay. In case of F value, so this value is equivalent to time in minutes at 250 degrees Celsius Fahrenheit of all heat considered. So with respect to its capacity to destroy spores or vegetative cells of a particular organism, or F is the time in minutes required to destroy microorganisms in specified medium at 250 degrees Celsius Fahrenheit. So these are different values. So the thermal death time curve, so mean viable count determined at intervals of 5 minutes. So these are below. So the time variable count, it is for see, here are different time variables they are given, and these are thermal date time curve is represented by this figure. So from this figure we can calculate d value. Okay. Then what is effect of pasteurization? So positive effect of pasteurization are destruction of pathogenic microorganism to increase the safety of market milk for human consumption. So basically this technique of pasteurization is applied to milk as well as milk related other product, as we have studied in milk microbiology, and improved keeping quality inactivation of certain naturally occurring enzymes. Okay. Negative effects are certain performed products of microbial origin are not inactivated during pasteurization. Example: staphylococcal toxins and aflatoxins. There is small loss of native aroma, particularly in case of fruit juices, and in case of milk, it destroys natural microbial property of milk by inactivating different natural occurring antimicrobial substances, and renate coagulation time is also increases. So these are pasteurization treatment for different foods. So here different foods they are listed. Main purpose of this technique is mentioned here, and subsidiary purpose is again mentioned, and what are the conditions of pasteurization they are given. So first, in case of fruit juice, so the main purpose is inactivation of enzymes, they are present in fruit juice. So pasteurization is carried out basically to inactivate enzymes which are present in fruit juices, and again destruction of spoilage causing microorganism, and these are different types of temperature and time combinations here we can use for treatment of fruit as well as fruit juices. So in that, 65 degrees Celsius for 30 minutes, then 77 degrees Celsius for 1 minute, and 88 degrees Celsius for 15 minutes. So these are different combinations of time and temperature are used here for pasteurization of fruit juices. Then in case of beer, it is basically used to destruction of spoilage organism, and these are combinations, say for example 65 to 68 degree Celsius for 20 minutes, and 72 to 75 degrees Celsius for 1 to 4 minutes. Then in case of milk, it used for destruction of pathogens and destruction of spoilage causing microbial organisms and their enzymes, and these are combinations, say for example 6 to 68 degrees Celsius for 30 minutes, and 71.7 degrees Celsius for 15 seconds. Then in case of liquids, destruction of pathogens and destruction of spoilage causing microorganisms achieved by pasteurization, and again these are different combinations of temperature and time are used to carry out destruction. Then in case of ice cream, destruction of pathogens and destruction of spoilage causing microorganisms. So these are different temperatures and time combination they are used. Then next, blanching. So blanching is a kind of pasteurization generally applied to fruit as well as vegetables and primarily to inactivate natural food enzymes. Okay, so blanching is what we apply. Blanching, it is used for treatment of fruit as well as vegetables. Okay, and it primarily what it does, inactivates enzyme, or it destroys enzymes present in natural food, and it is a common practice when such a foods products are to be frozen. So since frozen storage itself would not completely arrest enzyme activity. So basically blanching is used to inactivate enzymes, and peroxidase and catalase are the most heat resistant enzymes, and the activity of these enzymes is used to evaluate effectiveness of blanching treatment. So if both are inactivated, then it can be assumed that other significant enzymes also they are inactivated. So basically peroxidase and catalase, they are used as indicator. If these two are destroyed or they get inactivated, obviously others they are definitely get inactivated. So the heating time depends on types of fruit or vegetables and method of heating, size of fruit or vegetables, of temperature of heating medium. So these are different vegetables. Say for example bean, it can be means treated for 200 and 12 degree Fahrenheit for 1 to 3 minutes. Then beets, they are treated for 3 minutes. Then corn, they are treated for 2 to 3 minutes. Then peas, they are treated for 1 to 2 minutes. Then spinach, it is up to for 1 to 2 minutes. Then asparagus, it is for 2 to 12 minutes. So these are combinations at fixed temperature but varying time. Here temperature is fixed and the time of treatment is it varies. So rapid changes in color. So what occurs after blanching? So rapid changes in color, then flavor and nutritive value occur as a result of enzyme activity. Okay, so these are basic changes we found after blanching treatment. So it changes in color, we can observe, then flavor also it get altered, and nutritive value they get changed. So blanching is slight heat treatment, and using hot water or steam that applied mostly to vegetables before canning or freezing, and the main objective of blanching are to inactivate enzyme and to remove tissue gases, or to clean the tissue. So to increase the temperature of food, blanching is also used before canning for different reasons, and because enzymes will inevitably destroyed during canning, so blanching induces vacuum in canned goods, and it is also used to control into field containers, for example spinach. Then sterilization. So retorting. So sterilization means complete destruction or complete removal of all forms of life including viable endospores, and sterilization destroys all pathogenic and spoilage microorganisms in food and inactivates in them by heating. So all canned foods, they are sterilized in a retort, means a larger pressure cooker, and called as commercial sterilization, which indicates there is no viable organism are present. So in this process no organism is present after sterilization process, and this process enables food to have shelf life for of more than 2 years. So these kind of treated foods we can keep for 2 years. So this is pretty much a longer time we can keep. And foods that are have low pH, foods that have a pH of more than 4.6 such as meat and most vegetables must undergo severe heating conditions to destroy all pathogens. So these foods are heated under pressure to 121 degrees Celsius for varying times. So simply, so this principle of sterilization is a principle based on autoclave. So here foods are heated to under pressure, means this works under pressure, means steam under pressure is the basic principle used for treatment of in this kind of procedure, and severe conditions are applied primarily to ensure clostridium botulinum spores they are destroyed during this processing. So the spores produce the deadly botulinum toxins under anaerobic conditions, that is where there is no oxygen, and spores are destroyed by heating are inhibited at pH values less than 4.6, and therefore a food with pH less than 4.6 is packed anaerobically, such as spaghetti, sausage, etc. So some of the foods don't need to undergo such a severe heat treatment, and the destruction of vegetative or spore forming organism and pathogen is a secondary objective of commercially sterilized foods. So here we have discussed sterilization. Then canning is another term which is used for packed foods, and again we provide heat treatment for canning of foods. So here different kinds of canning treatment they are given. Say for example water bath canning. In this method, sometimes referred as hot water canning, uses large kettle or boiling water, filled jars to submerge with water and heated at temperature 212 degree Fahrenheit for a specific period. So here you can see hot water canning or water bath canning. Then pressure canning, similarly this operates as an autoclave, and steam under pressure is applied for pressure canning. So then drying is the another method of preservation of food. So here we simply dry foods, and after drying the moisture content get evaporated, and we can keep this food for a longer time. So one of the oldest method of food preservation is by drying, which reduces water activity sufficiently to delay or prevent bacterial growth. So drying is done to produce a concentrated form of foods, inhibits microbial growth and autolytic enzymes, and retains most of nutrient. So what we do drying, we kept for temperature, means open temperature, and we allows to evaporate the water from that food or any other food commodities. So drying can cause loss of some nutrients, particularly thiamine and vitamin C, and sulfur dioxide is sometimes added to dried foods to retain vitamin C, but some individuals are sensitive to these substances. So most types of meat can be dried, and this is especially valuable in case of meat. Then since it is difficult to keep without preservation, many of the fruit also can be dried. For example, the process is often applied to apple, pears, bananas, mangoes, papayas, and coconut and grapes. So we have you have seen that most of the time we dry fruits and we consume after drying that foods. Drying is also the normal means of preservation of cereals, grains such as wheat, maize, oat, barley, rice, millet, as well as rye. So in case of millets and grains also, we dry them in open environment so that to remove moisture from that grains, and we can keep this grain for longer time. So drying is an excellent way of preserving several of seasonal fruits using the off-season. Then there are several types of dryers which are used. These includes a drum dryer, then cabinet dryer, then tunnel dryer, then rotatory dryer, spray dryer, solar dryer. So all these are types of dryer that are used for different purposes as per the requirement of different foods. And the basic method of drying involves air contact drying under atmospheric pressure. So in this case, the heat is transferred through the food either from heated air or heated surfaces, and the resulting water vapor is removed with the air current. Solar drying and sun drying, drum drying, spray drying are all use this technique. Usually in our home we carry sun drying, means drying of foods and other grains as well as cereals in normal or natural temperature or natural heat under sun or under normal daylight or temperature. Then what are the advantages of drying? So it can means after drying we can keep foods for longer time. So long shelf like, then reduced weight, then convenience and concentration of nutrient, and so these are some advantages of drying, that we can keep dried food for longer time. So most of the microorganisms responsible for spoilage, they are unable to grow and multiply in absence of moisture. So spoilage due to microbial degradation is limited in dried foods. Then reduced weight, so this results in resulted transportation, storage, as well as shipping cost. Okay, so these are different advantages and disadvantages of drying. Then next, microwave sterilization. In this particular type, we use microwave waves for sterilization of different kinds of foods. So in these various kinds of microwave combinations, we here we use the radiation between different frequencies that they are used, that is up to 300 megahertz to 300 gigahertz. So due to this radiation, microwave radiation, most of the microbes they get killed. So this kind of treatment they are used for specific foods, and we can expect longer shelf life after microwave sterilization. So here you can see microwave sterilization, and apparatus used for sterilization is known as microwave. Okay, so this can be used for food industry, then including various kinds of frozen foods are sterilized by using this temperature. So here all the details regarding food preservation by thermal processing treatment. So in next lecture we'll continue with physical methods that are non-thermal origin, and in that we'll going to see different techniques of food preservation. Till that, thanks.